This video will cover subtopic 11.1, antibody production. Here are a list of understandings that we'll cover in this video, as well as some guidance and applications as well. So we'll start with the fact that every organism has unique molecules on the surface of its cell, and how this gets used by our immune system is basically that any foreign molecule that can trigger an immune response is referred to as an antigen. Now, the most common types of antigens are proteins and very large polysaccharides. Keep in mind that our own cells contain proteins and polypeptides and polysaccharides as well on the cell surface. And so this will help recognize between foreign antigens and self. So the fact that we have our own proteins and unique polysaccharides on our cell surfaces can help differentiate and distinguish between what is self and not self. So your immune system has to look to respond to not self and not respond to self. That's one of the ways in which the immune system looks to function. So an organism recognizes surface molecules present on its own cells, therefore unfamiliar surface molecules are regarded as foreign and will lead to a specific immune response. Now those specific immune response, in this case, that we'll focus in on, will be in the form of antibody production. So the analogy here is, think about answering the door at a party. You see your friends at the door and know they are safe, thus they are welcome, so you recognize them, you're like, you guys are good. Whereas if some stranger knocks on the door, you tend to proceed with more caution. So anything that you don't recognize or are not familiar with, you exercise caution. So unfamiliar surface molecules will cause the production of antibodies, these unfamiliar surface molecules are referred to as antigens. Now, antigens can take the form of like these surface proteins on bacteria, or the surface proteins on the outer coat of a virus. Now, with the novel coronavirus that's out there, one of the surface antigens is referred to as the spike proteins, which helps the virus bind to cell surfaces, but is recognized potentially as an antigen by our immune system. Now, antigens can be anything and anywhere. Anything that's unfamiliar to your immune system can act as an antigen. So in blood transfusions, if those blood cells being transfused into the body are not recognized by the immune system, they can act as antigens as well. So antigens on the surface of red blood cells will stimulate antibody production in a person with a different blood group. Blood groups are based on the presence or absence of certain types of antigens on the surface of red blood cells. And this is why we have 4 different blood types. So we have 3 different antigens, which are arranged in 4 different combinations. So we have antigen A, antigen B, and, well, there's no O antigen, it's just a lack of antigen. But it's basically the antigen A and B are in charge of deciding what blood type we have and what we can receive. So this diagram here just goes through the different ABO blood groups. So if you only have antigen A on the surface of your red blood cell, you have blood type A. Now, if this particular blood type were to be transferred into somebody who is blood type B, they would recognize this as foreign. But within the blood of that person now who has blood type A, they would have antibodies that would be against the antigen B. So they would have anti-B antibodies. So they would react against antigen B. Whereas somebody who is blood type B would only have antigen B on their surface of their red blood cells. Somebody who is type AB has both antigen A and B on their cell surface. And so in their blood, they would have neither antibody, so they would not have anti-A antibody, they would not have anti-B antibody. And then somebody who has neither antigen A or antigen B on their red blood cell surface, so they have neither antigen present, are referred to as type O. And these people will have antibodies, both types of antibodies, circulating in their blood. So what happens when a recipient is given the wrong type of blood? Basically, a reaction occurs that is referred to as agglutination. So what ultimately happens is the immune response in which, so it's an immune response in which the red blood cells are destroyed, and you can get coagulation that may occur within blood vessels. But ultimately what happens is, if you have those red blood cells coming in that have the wrong type of surface antigen. So this person here has antibodies that would recognize these proteins here on the cell surface. These antibodies are going to bind to that cell surface, and this will cause this agglutination to occur. And agglutination is just a fancy way of saying clumping. So you get this big clump of red blood cells, which will ultimately lead to hemolysis, those red blood cells being lysed and destroyed. So ultimately, those red blood cells that were transfused into that person will get destroyed. So now, dealing with pathogens and antigens here, and pathogens in relation to the immune system. So pathogens can be species specific, although they can cross species barriers. And pathogens are agents that cause disease. So it's any type of microorganism that will cause a disease. And so it's microorganism in the form of bacteria, protists, fungi, like fungal infections, think like athlete's foot, or a parasite, so think like tapeworms. Viruses can also be pathogens as well. And then you can have prions, which is essentially kind of a virus that does not contain nucleic acid. A prion is basically just an infectious protein. So a protein that can self-replicate and can ultimately lead to tissue damage and cell damage and eventually death. Mad cow disease is that classic example of a prion, a disease caused by a prion. And so pathogens are typically species specific in that their capacity to cause disease, when a pathogen causes disease, this is called pathogenesis, is limited to a particular species. So if you have a particular type of virus, let's say, it is specific to a certain species and typically does not cause infection in different species. However, certain pathogens can cross species barriers, and then you can get diseases from animals that are transmitted to humans. And when this happens, this is referred to as a zoonotic disease or zoonosis. And classic examples of diseases or pathogens that cross these species barriers include rabies, bird flu, and bubonic plague. So this diagram here just illustrates the fact that you can have diseases that occur in animals, and they can be transmitted to humans, and this process is referred to as zoonosis. So that novel coronavirus is thought to be a zoonotic disease, where its animal of origins is still under debate. But it was a virus that infected one particular host, and then there was some type of variation or mutant in that virus that allowed it to basically jump that species barrier and infect humans, and has kind of established itself in the human population. This diagram just kind of goes through the different zoonotic diseases that exist and the ways that zoonosis can occur. So it can be through wildlife and that interaction with humans. So Lyme disease is one example. Hantavirus, which is found in mouse droppings. Ebola is kind of the big one, where the host species is still not known, but it's a virus that's capable of infecting humans. And SARS as well, which is another type of coronavirus. Had its origins in wildlife and crossed over into humans. And then you can have zoonotic diseases that originate in livestock as well. So in certain parts of the world, humans live in very close proximity to their livestock, and so this can allow pathogens to go from one species to another. So one of the big examples of that is E. coli infection, particularly from cows, for example. So when you have E. coli contamination, that usually results from cows. And then you can have diseases that are kind of in between, that can go from wildlife to livestock to humans, or livestock to wildlife to humans. And the examples there are rabies and West Nile and tuberculosis as examples. And influenza is another example where it can infect things like birds and swine, pigs, but also infect humans. And it's one of the reasons why these occasionally you get these dangerous strains of influenza popping up. You get a various strain that might have been specific to a pig or a chicken, able to make the jump into humans, and it's a form of the virus that our immune systems have never seen before, and so it can make it potentially dangerous as well. So in looking at the specific immune response that gets generated, we'll look at this model of what's called the challenge and response in looking at how a specific immune response is generated. So the challenge and response explains how immune systems produce large amounts of specific antibodies needed to fight an infection rather than any other type of antibody. So when you get infected, or when you get an infection, your body will start to produce antibodies, and antibodies that are specific to that particular pathogen. And so what the challenge and response model looks to explain is how that one type of antibody gets generated to that particular pathogen and not other things. So antigens on the surface of pathogens are considered the challenge. So when you encounter the antigens of that pathogen, this is now a challenge to your immune response, and then your immune response is like challenge accepted. And then the response now will involve a series of processes occurring. So the pathogens are going to be ingested by macrophage in that process of endocytosis, which is phagocytosis in this case, but it's kind of that similar process of endocytosis. Now, antigens from the pathogen are displayed on the plasma membrane of the macrophage. So the macrophage will take in that pathogen, it will break it down, and it will take those little pieces and put them onto its cell surface. So it will present the antigen to the immune cells. Now, you have lymphocytes, these specific white blood cells called helper T cells, and they have this antibody-like receptor protein on their plasma membrane. The specific T cells that are able to bind the antigen are activated by the macrophages. So you'll have different T cells that will come along and interact with the antigen on the macrophage, and they'll interact with that antigen through their receptor protein. If that receptor protein is able to bind to that antigen on the surface of the macrophage, this will lead to activation of that cell. That cell will say, aha, this is my antigen, this is, my thing here. Let me let me become activated to be able to respond to this specific antigen. So the specific T cells that are able to bind to the antigen are activated by the macrophages. Once the T cells are activated, they bind to lymphocytes called B cells. And only B cells with specific receptor protein to which the binder antigen binds to are selected and undergo this binding process. So once the T cells become activated, they will then go and interact with a different group of lymphocytes called B cells, which have their own kind of antibody-like receptor on their cell surface. And if their antibody-like receptor is specific to that antigen, they will become activated. So very complicated process. This diagram tries to simplify this process here. So here we have the antigen presentation. I know it says dendritic cell, but dendritic cell is a different type of immune cell, but we can pretend this is a macrophage. It'll do pretty much the same thing. So the macrophage will eat that bacteria. So the little green thing is the bacteria here being taken up by phagocytosis. The bacteria, once inside the cell, that endosome will fuse with a lysosome. It'll break down the bacteria. And then the contents within that lysosome will be processed, and the protein portions will be displayed or moved to the cell surface, to the cell membrane, and be presented on the cell membrane. Now, this antigen that is presented here is just waiting for a helper T cell to come along. This is a CD4 T cell to come along. They have their own receptors, and each T cell will have a unique receptor. This particular T cell here has its unique receptor that is able to recognize this particular antigen. There will be other helper T cells that may come along, and they do not recognize this antigen. Because this helper T cell recognizes this particular antigen, it now becomes activated. It's like, hey, this is my thing here. Let me get going and start doing my job. So concurrently, you'll also have B cells around. They will have their own antibody-like receptors that may recognize a particular antigen on the B cell. And when they do, they'll be like, okay, this one fits, or, my receptor, fits this particular type of antigen. The other types of antigens that might be on these cells, I may not recognize, but I recognize this one at least. And so the helper T cell will come along and help activate that B cell. And this B cell now that's saying like, hey, I recognize one of these antigens, and helper T cell is like, cool, let me help you get activated here. And when that happens, this B cell will start producing antibody specific to this antigen. So once that happens, the activated B cell will now divide many times by mitosis. So that B cell, to help produce lots of antibody, because it knows that antibody will react to that particular antigen, will divide many times by mitosis, generating clones of that particular cell. And when a B cell becomes activated, it is now referred to as a plasma cell. So it generates clones of plasma cells that all produce the same antibody type. Plasma cells are mature B lymphocytes that will produce and secrete large numbers of antibodies during an immune response. So your body wants to produce lots of this antibody to be able to help eliminate this particular pathogen. So by undergoing mitosis, it ensures that it makes clones, which are all producing antibodies. And so generation of a large number of plasma cells that produce a specific antibody type is known as clonal selection. Basically, this one B lymphocyte has been selected for to undergo mitosis, and when it undergoes mitosis, it is essentially producing a clone of itself. So it's it's been selected to clone itself, clonal selection. These active plasma cells secrete antibodies, which will help destroy the pathogen. Plasma cells, once the infection is cleared, if we've managed to clear the infection, we'll assume that we have cleared the infection. Once that infection has been cleared, we don't need all those plasma cells now. So they are eventually lost. They undergo apoptosis and, self-select and remove themselves. And so a lot of those plasma cells get lost. But some of the clone B cells stick around, and they usually will stick around in your spleen, and will become what is called memory cells, and they will remain long after the infection is gone. So they stay somewhat inactive unless the same pathogen infects the body. They stay somewhat inactive in that they sit in the spleen and they secrete low amounts of that antibody that remains circulating in the plasma. And should you encounter that pathogen again, you have that antibody and you have those cells ready to go. So that antibody will bind to that antigen, and the B cells that are sitting there in the spleen will ramp up very quickly. So you can get that specific immune response to clear out that infection a lot faster. So antibodies now, we've talked a lot about antibodies in this immune response, but we'll talk specifically about what antibodies are and how they do their job. So antibodies aid in the destruction of pathogens. They are large, Y-shaped protein molecules produced mainly by plasma cells. And so the way that they aid in the destruction of the pathogens occurs through multiple ways, through this process called opsonization, which makes a pathogen more recognizable to phagocytes, so they are easily engulfed. Basically, an antibody will help macrophages take up those pathogens and help them destroy those pathogens. So you can get neutralization of a virus and bacteria. Um, so basically, what that'll do is, it'll have antibodies that dock to host cells so they cannot enter cells. But what it'll do is it'll bind to those surface antigens on a virus or bacteria that would be important for that bacteria or virus sticking to the cell surface. So if the bacteria or virus cannot stick to the cell surface, they cannot cause infection. So those antibodies are called neutralizing antibodies because they prevent the bacteria or the virus from doing their thing, so they neutralize. Um, Antibodies can also neutralize toxins. So some antibodies can bind to toxins produced by by pathogens, preventing them from affecting susceptible cells. And then you can also get this activation of complement. So there are these proteins, this protein system called complement proteins, which can attach to a pathogen, causing it basically to lyse. So antibodies will bind to the pathogen, to the surface of a bacteria, for example, and will occur like these proteins, these complement proteins, to attach to the pathogen, causing it to lyse eventually. And so this diagram just goes through the different roles or the different methods in which antibodies help remove pathogens. So if you think panic, you have precipitation, agglutination, neutralization, inflammation, and complement. The different ways in which antibodies work. With agglutination, which is basically the kind of the form of precipitation, antibodies will attach to each other, causing clumping of the pathogen. What this does is it enhances the effect of neutralization or opsonization. Basically, it'll enhance that engulfing or that phagocytosis by macrophages. So rather than, kind of taking up one thing at a time, a macrophage can take up many pathogens at one time. So that's kind of the immune response in a nutshell. And then what we'll kind of look at are variations or ways in which our immune system may not react in a way we would want. And this gets into the idea of allergies. And then we'll have a look at how we can manipulate the immune response to protect us against certain pathogens, which gets into this idea of vaccination. And then we'll look at how antibodies are also used diagnostically and medically as well. So we'll start with this idea of allergies here. And we'll start with this idea of an allergen. So an allergen is basically an environmental substance that will trigger an immune response despite not being harmful. So it's an innocuous substance out in our environment that our immune system is responding to. It thinks it's a threat, but it's not a threat. So the immune response that is localized to the region of exposure is called an allergic reaction. Severe systemic allergic reaction is called anaphylaxis and can be fatal. So with an allergic reaction, it does require a prior exposure to an allergen, and specific B cells first encounter the allergen, differentiate into plasma cells, and make large quantities of antibody, typical immune response. The antibodies attach to a particular type of immune cell called a mast cell, which is found in the connective tissues, priming them towards the allergen. So the antibodies attach to the cell surface of the mast cells. The mast cells sit in that connective tissue, and they basically wait for antigen to attach to those antibodies. Now, upon re-exposure to that allergen, the mast cells will become activated and will release large amounts of histamine, which cause inflammation. So it's those mast cells that release histamine that cause those allergic reactions. So this diagram just runs through how sensitization will occur and how an allergic reaction occurs. So you have a flower here that gives off, its pollen, which we'll say is an allergen. It has like little proteins on its surface that our immune system might recognize as being harmful, but it's not actually harmful. Pollen isn't harmful. We generate an immune response to it, so we generate antibodies to it, and we generate a particular class of antibodies referred to as IgE antibodies. Now, there are different classes of antibodies that are produced, and those antibodies are found in different areas. So there's a type of antibody called IgA, which is found in mucosal surfaces. So like in your nose or in your immune system is where you'll find a lot of IgA. The antibodies circulating in your plasma or your blood is another class called IgG. And then IgE, this particular class of antibody, will bind itself to the surface of the immune cell referred to as a mast cell. Mast cells contain contain large amounts of histamine within them, and so you'll have below your skin surface or like in your nose or something, large amounts of mast cells just sitting there with that IgE antibody, waiting for that antigen to bind to the antibody. So now the allergic reaction will occur. So you have that antigen, that pollen that you breathe in, binding to those IgE antibodies on the surface of the mast cell. The mast cell is like, all right, this is it. We got to go into action now. Let me do my thing. And so it will now release all that histamine contained within it. And you got to think there's a lot of mast cells doing this. And then the histamine is what will cause that allergic reaction. So it causes your eyes to water, your nose to run, cause you to sneeze. These various kind of basic defense mechanisms to try and like flush out thing that your body thinks shouldn't be there. So histamine is what causes those allergic symptoms, so it'll also cause an inflammatory response. So it'll help improve leukocyte or lymphocyte mobility to infected areas by triggering vasodilation. So what histamine will also do is it'll cause your blood vessels in that localized area to widen for better circulation, but it'll also help increase capillary permeability. So it'll kind of increase the leakiness, I guess, of those blood vessels as well. So it'll cause those leukocytes to more readily be able to leave the bloodstream and migrate into the body tissues. Basically, it's going to allow those leukocytes traveling in the blood to say like, there's some holes for me to escape, and then will leave the blood vessel into the tissue. And then this can lead to swelling and pain. So this figure here just kind of looks at this process of immunity and kind of how long it takes things to happen. So immunity, this idea of immunity is due to either the presence of antibodies that recognize antigens associated with disease, or memory cells that allow production of antibodies. So the way that immunity comes about to a particular pathogen, if we look at kind of the level of antibody concentration in the blood, is that, upon first exposure, you have very low levels or no levels of that antibody. And it takes almost about a week or so to kind of get a peak exposure. So after first exposure to pathogen, it takes about a week to kind of reach that initial levels. And then those levels kind of linger around for a while. And then at some time in the future, so they go back down pretty low, so at some time in the future, if there's a second exposure to the pathogen, you get a more amplified antibody response, and an antibody response that's generated a lot quicker. So here we can see the peak antibody response occurs 3 days after the second exposure to that pathogen, whereas the peak response is occurring about 5 days out, and typically it takes a bit longer to reach that peak upon first exposure. 5 days is a bit quick. This video here, which we won't do a video within video, goes through this whole process of an immune response that has been discussed from initial exposure all the way up to a memory response or a second exposure. So I'll provide a link to that video in the description if you want to check out this 2-minute video that summarizes what has taken me about 30 minutes at this point to go through. So the other thing that we'll touch on now is this idea of vaccines. And this is kind of using our immune system, or priming our immune system to protect us against certain types of pathogens. So what a vaccine is an attenuated form of a pathogen. And when we say attenuated, we mean weakened form of the pathogen that contains antigens but does not cause the disease. So it can be attenuated in that, it's the pathogen could be primed to infect a different type of species cells. So influenza vaccines, for example, are grown, that influenza virus is grown in chicken eggs, which weakens its response in humans, as it's kind of more geared towards chickens. Or we can heat kill a pathogen, or you can inactivate it in a preservative. There's different ways of attenuating that pathogen. So when vaccines are injected or taken orally, there aren't too many oral vaccines. Most vaccines are injected, as you may be well aware. It will initiate a primary immune response. And the ultimate result is that it results in memory cells being produced. So it'll go through that process now of T cell activation, which leads to B cell activation, and then you'll get B cells producing that specific antibody, and then those plasma cells will eventually become memory cells, and it's those memory cells that are going to provide us with long-term protection should we ever be exposed to that particular pathogen. When actual pathogen is encountered, those memory cells are triggered, leading to that secondary immune response. And you saw from the previous diagram that when there is a secondary immune response or secondary exposure to a pathogen, that immune response is greater, or the production of those specific antibodies are significantly higher than the first exposure. So the secondary immune response is much faster and stronger, and thus little or no symptoms are experienced. The length of time a person is immune to infection depends on how long the memory cells survive. So booster shots may be needed every now and then, and you may have to go and get booster shots for certain vaccines every so often. So again, this diagram here shows what an immune response, or what that antibody response looks like over time. So here a vaccine has been provided. Here's the primary immune response. So it's low, it's slow. And then here is where we've now generated some memory cells. And then later on, at some point in your life, you may encounter that pathogen that you have been vaccinated against. And that secondary response is a lot stronger and more rapid than that primary response. So you generate a lot more antibodies, you can clear that pathogen a lot more quickly, a lot more efficiently, and so it is less deadly in this sense than if there were no vaccine provided. So smallpox was a disease, and the has several distinctions. One of the first diseases in which vaccine was created. In fact, the word vaccine comes from the term vaccinia, which is Latin for cow. And it was cowpox that was kind of used to vaccinate people against smallpox. Vaccination always, kind of comes back to smallpox. So smallpox, was the first disease in which a vaccine was created. And smallpox was also the first infectious disease to be eradicated through vaccination. It's kind of, circular in a way, or kind of closes that loop that, it was the first disease for which a vaccine was created, and, the first disease to be eradicated through vaccination. The last known case globally was in 1977, and the immunity was long-term, so booster shots were actually not needed. This diagram here shows the number of countries reporting cases of smallpox. And in 1967, this is when the eradication program began by the World Health Organization, and they undertook a global effort to vaccinate like everybody on the planet and track down the disease. They tracked down this disease to the corners of the earth, until they identified like the last known smallpox case, and once that had been done and cleared, and there were no more reported cases, they declared smallpox eradicated. So there has not been a smallpox case since 1977, but smallpox still does exist in test tubes in a few labs around the world in freezers. So it's been eradicated, but not completely gone. So with this idea of epidemiology and tracking diseases, this leads into the idea of epidemiology. So epidemiology is the study of the incidence, distribution, and possibly control of various diseases. It can be used to compare incidence of diseases over time, and can be used to compare different regions. During this whole coronavirus outbreak, Tracking the disease has been the job of epidemiologists, and modeling like the various infections. Epidemiologists have done that as well. So the last thing that we'll get into is this use of antibodies for medical uses, so either therapeutically or diagnostically. And the use of antibodies will be based on this idea of what's called monoclonal antibodies. So if you think back to this earlier in the presentation about this idea of clonal selection for a plasma cell, so that is a B cell that produces a specific type of antibody. This is the idea of monoclonal antibodies is based on this idea of clonal selection. So if we can identify a single B cell clone that is producing an antibody of interest, we can basically take that single antibody or that single plasma cell, get it to create clones of itself, and then we can use those plasma cells to produce large amounts of that specific antibody, and harvest that antibody and use it for what we need to. So a monoclonal antibody is referring to just basically a single specific antibody that where you have antibody, or you basically have a solution of antibody that reacts only to one type of antigen. So monoclonal antibodies are artificially taken from a single B cell clone. The way in which they're produced is that you have an animal, in this case a mouse, injected with antigen, and produces antigen-specific plasma cells. Those plasma cells are taken out of the mouse and will be fused with tumor cells capable of endless division. So these plasma cells are limited in their capability of dividing, whereas tumor cells have basically unlimited cell division capacity. So you take these tumor cells, you fuse them with these plasma cells, and you create what are called hybridoma cells. So they are a hybrid of tumor cell and plasma cell. So you kind of get the best of both worlds here in these hybridoma cells. You get these cells that divide endlessly and crank out your antibody of interest. So the resulting hybridoma cells are capable of synthesizing large quantities of that monoclonal antibody. And so monoclonal antibodies, like I said, have various uses. So they can be used therapeutically. So one example is in the treatment of rabies. So rabies can be fatal, so thus injecting purified antibodies will act as an effective emergency treatment. So those antibodies will bind to that rabies virus, helping prevent disease caused by that virus. Now, antibodies are also used in medical diagnosis, so they're used in detection of pregnancy. Pregnancy kits or pregnancy sticks or whatever you want to call them make use of monoclonal antibodies. And so the way they work is those antibodies are specific and will detect the presence of the human chorionic gonadotrophin in urine. So this HCG, which indicates pregnancy, and it can detect levels of that within the urine. And where antibodies also get used in medical diagnosis is in this particular type of test called an ELISA. And ELISA is basically an acronym for Enzyme Linked Immunosorbent Assay, which is used to test for specific substances. And it uses kind of a color change here as the assay part of it. We won't go into the specifics of what all that means, but it's basically a test that shows whether something is present or not through a color change, and makes use of antibodies. Having a look at a pregnancy test, though, and where antibodies get used. So if this is the stick here, so this is the reaction site. So this is like the absorbent strip here. This is what gets peed on when you're testing, wondering if a pregnancy might be possible or not. And so there's a few things going on here. So there is the test site, and then there's the control site on the stick. So basically, once the urine is on here, it will wick. The pad here acts as a wick, drawing the urine up into the test site. So basically, on the reaction site, um, on this pad here are these free antibodies that are specific to the HCG. Now, these antibodies contain a dye and an enzyme attached to them. So if the HCG is present, it'll bind to this antibody. That free antibody will wick up into the test site. So at the test site, you basically have this particular type of antibody here that is now fixed in this test site area. This will trap this whole complex here. And it will, and if it does trap anything here, um, or it'll trap the HCG here. If it does trap that, you'll get a reaction occurs that will come up as a line. And then there is also a control site here, which proves the test is working. And this fixed antibody here will trap this free antibody. And will activate. So it'll trap this antibody here, and you'll get a line that shows up here. So it shows that the kit is still working, or the test is still working. So you always get a line here. But if the test is positive, meaning there's presence of HCG in the urine, you'll get a line here. If there's no HCG, you won't get a line here, but you'll still get a line at the control site. And so ultimately, the pregnancy test here makes use of monoclonal antibodies, and the idea that antibodies can bind specific antigens, and we can develop tests around it for various things. So that ends this video for 11.1.